Well, thank you again. And my name is Eva Lundgren Åkerlund. I am the CEO of Xintela and also the founder of the company. I also have Thomas Areskog here, sitting down there, our business developer, and actually also the CFO, Gunnar Telhammar, here today. So we are located at Medicon Village in Lund. We have a team a fantastic team of 17 people working really hard to reach our goals. And we are listed on Nasdaq, First North, Stockholm. That picture is the site of Medicon Village in Lund. Very active life science site. Also have a fantastic board with broad experience in pharmaceutical drug development. Extra happy to have Sven Kiely on board with a really long experience in developing cell therapy products from Genzyme and Sanofi. But also Greg Batchelor, Claes Post, Conny Wingstrand has great experience from both biotech and the pharma industry. So our business, we develop products in regenerative medicine, in cell therapy, and in cancer. And our focus is osteoarthritis, the joint disease osteoarthritis. And in cancer, it's glioblastoma, this terrible brain tumor, glioblastoma. And our business is based on a patented marker technology platform, an integrin technology platform, which we call Xinmark. So, and our markers. So what is a marker? Well, our markers are proteins, cell surface proteins, present on the surface of specific cells. And here you see one of our integrins, the one we work mainly with, integrin alpha 10 beta 1. We discovered this integrin on chondrocytes. It's present on chondrocytes. We later on discovered that it's also present on stem cells. And a few years ago, we discovered this integrin on certain cancer cells. And we use this integrin or our integrin markers to commercialize products in regenerative medicine and cancer. And we have developed antibodies tools, protein tools that bind to the markers. And we can say that the integrins are sitting like recognition flags on certain cells. And then we have this integrin here. We developed antibody to that one. We have another marker that recognizes the fibroblast, which is different from this integrin that recognizes chondrocyte, cartilage cells, and stem cells. So when we culture these cells, they look very similar. You see here, difficult to distinguish between these cells. But with our integrin markers, we have a great way actually to differ a chondrocyte and a stem cell from a fibroblast. And we use these antibodies to do that. And these antibodies that we have developed, they work as like a tool to quality control chondrocytes and stem cells in cell therapy. And we have developed what we call EXACT, Xintela assay for cell therapy. An assay based on our antibodies to show identity of cells, to be able to differ between a chondrocyte and a fibroblast, determine purity of cell preparations, and also to predict the potency of the cells, so the function of the cells. And we use this technology in our own stem cell product development. But we also offer this technology to companies that work with chondrocytes, with cartilage cells, to develop product based on that. So an EXACT, so this assay, we have already generated some income through collaborations with other companies. So Codon in Germany, a collaboration that we have completed, and we have now an ongoing collaboration with the Japanese company Cellseed. And in both these companies work with cartilage cells, with chondrocytes. So the goal here for us is to out-license EXACT for quality control of chondrocytes. So in parallel with our own product development. So regenerative medicine, which is one of our main focus areas, or we develop stem cell products for cell therapy. We focus on the joint disease osteoarthritis. And in this disease, the cartilage is gradually degraded. It is very painful, debilitating for the patients. You can here in this drawing see that the cartilage has been degraded and the bone is exposed, making the pain. And osteoarthritis, huge growing problem, and a lot of people suffer from this disease. In Sweden, it is estimated that 25% of the population over 45 have osteoarthritis. It can happen in many different joints: foot, knee, hip. And the cartilage has a very bad ability to heal itself. And there is no treatment. Today we treat symptoms to relieve pain, to decrease inflammation, and many of the patients end up with a joint replacement surgery. Over 1 million joint replacements are done yearly in the US, in the EU. And of course, this is a large and growing market. And we aim to take a part of that market. We develop what is called off-the-shelf stem cells, so an off-the-shelf product, or actually we can say off the freezer, because our stem cells are produced and put into vials like these that are kept in the freezer, and in the clinic, the cells are thawed and aim to be injected into the joint to be treated. We use what is called allogeneic stem cells, so they're donated stem cells that we isolate from fat tissue, from adipose tissue. And one great advantage in working with donated tissue instead of the patient's own stem cells is that we can produce huge amount of stem cells from one batch, from one adipose tissue preparation. And that means that this is a very cost-effective production. We can develop a product at a low cost. And also the stem cells will be injected into the joint, so no surgery involved, and one-step procedure, an easy procedure for the patients. So how are we different? We are not the only stem cell companies developing products for osteoarthritis, but we do something that is different to the others. We actually select stem cells to get high-quality stem cell preparation. And we use our integrin target and our antibodies to do that. If you look at stem cell preparations from adipose tissue, they're quite heterogeneous. And this is a problem in developing a stem cell product. Stem cell products are very complex. And it's important to show that stem cell from donor to donor is the same, or is very, very similar. The problem is that they're heterogeneous. What we do, we use our marker that is present on stem cell, our antibodies to select, to get a homogeneous cell preparation. This has a great advantage. It has biological advantage because we know that the cells that we select with our technology, they have a high ability to develop into cartilage cells. We show that here. This is a pellet of stem cells that have our market on the surface, and they can differentiate to cartilage cells and start to produce the cartilage molecule, type 2 collagen, which is we here stain for. It becomes brown when we stain for that collagen type 2. But the stem cell that lack our marker does not have this differentiation potential. So great biological advantage. So we have a great base here to develop a safe and effective stem cell therapy to stop further development of osteoarthritis in a patient, and even help the cartilage to heal, and also to be able to reduce the need for joint replacement surgery. We have evaluated our selected stem cells in horses. in preclinical studies in horses. The horse is a great model for these studies. It's actually a preferred animal model for clinical studies in human. And also it's a great business. It's a large need, of course, for therapy also for horses with cartilage damages or cartilage damage. So, and another great advantage is that the stem cell product for horse has a shorter time to the market. So we work with both human and horse. Our selected stem cells, we selected the cells in our lab, put them into these vials, shipped them to US, to Lisa Fortier and to her team there, where our stem cells were evaluated in a horse model, where it's actually a model for post-traumatic osteoarthritis. So these horses, they had a damage in their ankle joint, their hook joint. So our stem cells were injected, 20 million stem cells into one joint, and a control, like a salt solution, into the other joint. 6 months after this treatment, the cells or the joints were analyzed. We looked at the cartilage bone. We also looked for safety, of course, in other tissues, and analyzed joint fluid samples. This is result from one of these horses, and we found, so this is the intact cartilage from the control and the treated joint. And this is where the damage was. So this is the control cartilage. You can see the damage. And we found that when our cells were injected after the damage in this joint, there was less structural damage in the cartilage, so less damage. That was one example, but we also investigated these in the 8 different horses. And we found significantly less cartilage fibrillation, so cartilage damage in the treated joints. So this was really, really good news. So we could conclude from this study that we got really positive results. The method also to inject stem cells, one single injection into a damaged joint, that is feasible. We could also conclude that the stem cells are safe and that they had a significant protective effect on the cartilage and actually also on the bone, so less bone sclerosis after the treatment. And interestingly, we also found some indication of repairing effect of these damages in the cartilage. If you have a chance, go to our website and look at the film where Lisa Fortier talked about this. The results from this study. I mentioned biological advantages, but our technology also has several regulatory advantages. And I talked about homogeneous preparation and that is very important. And also to have that we can have consistent quality between donors and batches. Also in process control during our production to control the cells. And also one advantage is that we can predict the function, the potency of the cell. So as I said, this is a complex product, but with our technology, we have a way to define the product and also keep it consistent. Great news that we came out with a few months ago is that we now have built our own GMP facility. So this is a facility to produce the stem cells for clinical studies. GMP facilities is a good manufacturing practice. So it's a GMP lab where we produce the cells under very strict regulations to be able to do clinical studies with these cells. Great advantage to have process development, manufacturing at the same site. And also, this really reduces the cost for us and to lower the manufacturing cost, even in short term. Another interesting opportunity here is that we can actually produce the cells also for our collaborators and as a contract manufacturer. So a great business opportunity here. So in the last sentence there, full control and flexibility, a great advantage for us. We have reached important milestones We have positive preclinical results in horses. We've been able to scale up our production into, in a bioreactor system that is now in the GMP lab, and we built our own GMP facility. A few months ago, we also came out with some news that we have the possibility also to enter into a completely different field with our technology. We have been able to show that our technology can be used to isolate and quality assure neural stem cells for the treatment of brain damages after trauma, after stroke, or for treatment of degenerative diseases such as MS. We will talk more about this when we have developed this further. Milestones for the stem cell project. So what's going to happen now the coming years? Well, we will start a new horse study this year to investigate dose and also mechanism of the stem cells. We'll also do a biodistribution and a toxicology study of human cells. We plan to have a manufacturing approval from Läkemedelsverket, Swedish MPA, before the end of this year. And we will also have a development strategy for the neural stem cell project, how to develop that, either in-house or in further development with a partner. Next year, we plan to start our clinical study. We plan to be able to do that in the end of 2019 on humans for the treatment of osteoarthritis. And also in parallel, we do preclinical evaluation of new indications for stem cell treatment that includes other joint diseases, but also damages and diseases in the brain. So with that, I'm going to switch over to the other project, oncology, where we develop new cancer therapy. And our focus is the brain tumor glioblastoma. Most common, most aggressive form of brain tumors in adult. Affect a lot of people. As I said, it's very aggressive. So you see the average survival without any treatment is only 4 months with this tumor. And with current treatment, it's only 15 months. So there is a large need for better treatment methods. And certainly an interesting market potential. That is also expected to grow when new treatments are available. We discovered some years ago that one of our markers is expressed on glioblastoma cells. And this showed us that with our technology, we have a great way here to develop a new treatment method. And this picture here shows it's a tissue from a glioblastoma tumor. And the brown staining are tumor cells that express our marker or our target in this case. So we have developed a new treatment method, a targeting antibody. So we use one of our antibodies that we have developed that bind to a target and that we know that bind to the glioblastoma cells. We combine this antibody with a cell toxin to develop an antibody drug conjugate, an ADC. And the goal with this treatment is to kill the tumor cells and to stop the tumor growth. We have positive preclinical results from the ADC. We have found a killing effect on glioblastoma cells, both in cell studies, but also in vivo models. So when we inject the antibodies into the animals with the glioblastoma tumor, we can see reduced or a reduced size or a dying, that the tumor cells are dying, so non-viable tumor cells. We have reached important milestones in this project. We have proof of principle in animal model for glioblastoma. Also a number of additional results in cell studies that support the concept. We've also licensed in human antibodies to develop an ADC based on a human antibody. And interestingly, we have also identified new indications in oncology. And with all these, the progress that we have done in this oncology program, we have found that it's now the right time to look at the possibility to spin out the oncology project into, or the program, into a separate company. So the milestones in the oncology projects, well, we plan to establish a collaboration for further development of the glioblastoma project. We will develop and optimize the ADC concept, and also importantly choose new oncology indications for the development. And we will also during the year decide on a possible spin-out of the oncology business. Next year will be further preclinical development of the ADC treatment for glioblastoma to prepare for clinical studies, and certainly also evaluate new oncology indications for our ADC concept. We have a continuous and very active process to identify development partners and licensees for our projects, for all our projects. In the stem cell project, we plan, our strategy is to find a development partner after proof of concept in humans, after our first in-man studies. On the veterinary side, for horses, also for dogs, our aim is to have a development partner after preclinical proof of concept. And with our horse data, we have already a big, major part of the preclinical program ready. In the oncology projects, our aim is to identify a partner, co-development partner, licensee after preclinical proof of concept. And as I mentioned, the plan is also to spin out the oncology business to a separate company. For Exact, our quality assay for chondrocyte preparations, we have ongoing discussions right now with potential partners or licensees. So my last two slides here are the take-home message, we may say that. In regenerative medicine, with our proprietary marker technology and our stem cell platform, this combined with our GMP manufacturing facility, we have a really strong and attractive position in the development of cell therapy products. In the oncology part, we have targets now shown to have large potential in oncology. First-in-class therapy, promising preclinical results, new cancer indication, which means a broader use of our ADC concept. And also the spin-out of the oncology program to a separate company will really, we believe, with a separate financing, separate management, this program and has larger opportunities to growth and will increase the value of the oncology program. So with that, thank you very much.
Thank you, Evi, for an interesting presentation. It sure seems that you have a lot going on at Xintela. The last big news was the inauguration of your GMP facility. This is a huge step for a small biotech company to have their own GMP approved. Well, it's not actually approved yet, I would say, but
that's right. But
to be able to produce cell material yourselves. What's in the big picture? What will this mean for Xintela?
It means a lot. And as you also say, it's not a common thing to do for a small company like Xintela. I mean, the major stem cell companies have their own, but it's really a big step forward with many advantages, as I said. Actually, the idea came when we looked for opportunities to produce horse stem cells, and it was really difficult to find a production facility that would take in the horse stem cells into their unit where they produce human stem cells. So that, with our stem cell facility, we can control that, and we can produce in campaigns. So we can during a few months produce horse stem cells and then clean out and then do a new campaign with human stem cells. So that's what we mean. We have full control, we have full flexibility. And also, this is, we're building really big value into the company. And we build up great competence, and with this facility, we can actually build a business around this. And when we discuss with potential partners, it's a great advantage for us to be able to produce in a collaboration, not only offer our stem cell technology, but also to offer the production ability of the stem cells. So this is a big step. Great news.
And in terms of, you mentioned you have had some revenue coming in from your collaborations. The GMP facility in itself could also be a source of income.
Yes.
If you partner and if you have external companies coming in to basically then rent your facility. Is that correct? Or?
How that will be handled, we'll see. But I mean, we have, it's now built to produce our stem cells selected with our technology, and that will be easy to produce stem cells for collaborating partners. And how this will develop, future will tell.
Yeah. Okay.
But the opportunities is certainly there.
Yeah,
Yes.
and the main focus is still your own material for your own upcoming studies and so forth.
Yes.
You also mentioned that the partnering strategy is to await the clinical proof of concept in humans, of course, for the osteoarthritis stem cell project. But then in the veterinary indications, then you're looking for preclinical proof of concept first. And you mentioned that you already have quite a lot of the preclinical package ready. What's missing, so to speak, before you can close that deal?
Yeah, well, actually, we have discussions already, because we have done a great deal of the preclinical part already with the study that we did at Cornell University, where we showed the great results with our stem cells. We are now starting a new study to look at the dose, to the optimal dose, and we are also looking at mechanism of action to get a even better understanding of the mechanism of action of our cells. But we have come quite a long way already with the horse study that we've already done. So that's what we'll do, is to prepare the stem cells even further for a clinical study in horses.
Yeah.
And I would also like to say that this, of course, is also preclinical data that we use in the preclinical package for human stem cell study.
There's a synergy effect there. What kind of partners are you looking at or discussing within the first, so to speak, stage where you're looking at the veterinary part? What kind of companies is there? The several big veterinarian company that are interested in stem cell technology. I guess you can't say more than that. I'm going to ask the audience if you have any questions for Evi at this point. Everyone, you had such a fulfilling presentation. There's no need to ask questions. What's coming up? What's the most important milestones coming up next for you? I know that the GMP facility needs some more validation. Is that correct?
Well, you were at our inauguration a few weeks ago, so the GMP facility built, the instrument that is in there, and now we're preparing finalizing all the optimization, the process, and the quality system to be able then to have everything in order to apply for an approval from Läkemedelsverket, from the MPA. And of course, a big step will be, a milestone will be when we have an approval to start producing. And our plan is to have that by the end of the year.
By the end of the year, okay. Yeah, that sounds great. Will you be hanging around here for the lunch, and then you will be able to ask questions?
Yes.
I would like to thank you, Eva, for coming today.
Thank you very much. Nice to be here.
Yeah.